Welcome to this webinar on Standards and Accreditation for Immune Effector Cell Therapy, brought to you by FACT, the Foundation for the Accreditation of Cell Therapy. I am Dr. Phyllis Workington, Professor of Pathology, Microbiology, and Pediatrics at the University of Nebraska Medical Center and the Chief Medical Officer for FACT. The objectives of this webinar are to enable the participant to describe the purpose and scope of FACT standards and accreditation, including immune effector cell therapies, to compare and contrast requirements of quality management and sponsor manufacturer-specific requirements, to discuss experiences of an accredited program implementing immune effector cell therapies, to list examples of multi-stakeholder standardization groups in which FACT participates, and to list services provided by FACT Consulting Services. FACT was founded in 1996 by ASTCT and ISCT to be the standard-setting and accreditation body for hematology oncology and stem cell transplantation. FACT was founded to address perceived need for minimal standardization across these fields, to mitigate perceived burden of regulatory oversight, to develop an accreditation process that comprehensively encompasses clinical practices, cell collection, and laboratory services, and to foster research and development in the field. FACT was originally founded in North America, but soon began collaboration with colleagues in Europe to form the Joint Accreditation Committee of ISCT and EBMT, or JC, to foster international collaboration. JC adopted the first edition of FACT standards in 1999 and has subsequently participated in the development of all editions of the hematopoietic cell therapy standards. In 1998, FACT also collaborated with NetCord to expand expertise in the field of cord blood banking and published standards for this field. FACT also partners with CIBMTR to improve data management and data auditing practices across the field. The goals of the FACT community today are to promote quality patient care, improve treatment outcomes, and foster continued development of the field. These goals are pursued through three core services: development of standards, awarding of accreditation, and the provision of education. All FACT standards are developed by consensus of experts in the field. They are unique in their depth and breadth. For the patient, standards cover all therapies across the continuum of care, including inpatient, outpatient, and ambulatory settings. They cover all phases of care from diagnosis or referral, all cell therapies, follow-up, and reporting of long-term data. FACT standards cover cellular therapy across all phases of manufacturing, from collection, simple processing, complex manufacturing, storage, and administration. For the transplant program or cellular therapy program, standards cover all facilities, including the clinical program, the collection, apheresis center, and processing facility or laboratory. FACT standards are based on published medical literature whenever available. FACT standards are always published for public comment and regulatory review, revised, and approved by the Board of Directors. FACT standards represent minimal requirements and are not intended to establish the best practices in the field. They are not prescriptive, but allow flexibility in that there are many ways in which a program can comply with the standards. All FACT standards require a comprehensive quality management program to be in place. They require assessment of relevant endpoints. These endpoints can be variable depending on the phase of product. For early phase products, safety and efficacy are the endpoints, whereas for later stage products and clinical trials, clinical patient outcomes are important. All standards also require compliance with regulations of the competent authorities in the jurisdiction of the program. FACT collaborates with JC, the Joint Accreditation Committee of ISCT and EBMT, for the development of hematopoietic standards and the immune effector cell standards that are embedded within this document. FACT standards requires an integrated medical team or program to administer cellular therapies. Importantly, the clinical program must use a collection and processing facility that meet FACT-JC standards. Likewise, the apheresis collection facility must use a cell processing facility that meets FACT standards. This one-way flow of standards requirements enables the clinicians to be ensured that all aspects of product manufacturing will be optimized for the care of the patient. The FACT accreditation program is based on the FACT-JC hematopoietic cell therapy standards. It's a voluntary process in which programs hold themselves to a higher standard for the benefit of patients. Accreditation is based on documented compliance with all standards as shown through submitted documents and an on-site inspection. The process is peer-driven. Inspectors are volunteer professionals active in the field and experts in the area they inspect. Inspectors teach, coach, mentor, and learn from these on-site inspections. The process is consistent as ensured by staff coordinators and a professional accreditation committee review of all findings of the application prior to the awarding of accreditation. FACT accreditation is not a one-time pass-fail event. There are opportunities for programs to make corrections and improvements throughout the process, with the overall goal being to help programs and cord blood banks improve their quality and achieve accreditation. The process balances a rigorous, comprehensive inspection with collegial approach, helpful inspectors, and dedicated support from FACT staff and volunteers. This timeline is to emphasize that the FACT accreditation process does not end with the on-site inspection. The on-site inspection, shown here on the bottom of the slide with the red arrow, occurs roughly 5 to 6 months into the application process. Following the inspection, inspectors prepare a comprehensive report of findings or citations. These are issues they found that showed a lack of compliance with a FACT standard. This report is reviewed by FACT staff coordinators and an accreditation committee comprised of experienced inspectors and board members. The final report lists each citation and is sent to the program. The program then has time to correct these deficiencies, document the corrections, and submit that documentation to FACT. These responses are reviewed, and if satisfactory, accreditation is awarded. The FACT accreditation program is international, having accredited 184 clinical cellular therapy programs in 7 countries. The collaboration with the JC is through standards development. The standards for hematopoietic cell therapy and the embedded immune effector cell standards are jointly developed and published by FACT and JC. They are approved by the boards of FACT and of EBMT. The accreditation programs are similar but completely independent. The similarities include a volunteer inspectorate, checklist documentation, and independent review by staff and an accreditation committee. There are differences in languages, in the cycle length of the accreditation, and in the structure and identification of the individual committee members who review the processes. FACT publishes 4 sets of standards. The most basic are the common standards, which are those requirements that apply to any cellular therapy. These standards can be used for fields not ready for specialized requirements or as the foundation for the development of new areas or products. The most mature standards are the hematopoietic cell therapy standards. These are common standards plus the unique requirements related to hematopoietic cell transplantation. Standards for immune effector cells are incorporated into these standards. The standalone immune effector cell standards are common standards plus those unique requirements related to immune effector cells. They lack any standards related to transplantation specifically. This arrangement allows an organization to use one set of standards relevant to its activities and increases transparency and clarity regarding the requirements under which an organization is accredited. The cord blood standards are also published independently. They have considerable overlap with the basic requirements seen in the common standards. Therefore, the set of standards applicable to any specific program depends on the services offered by that program. The hematopoietic transplant program that offers immune effector cell therapies will follow one set of standards. A standalone immune effector cell program could follow the immune effector cell standards alone, and new and developing translational cell therapies will follow the common standards. For purposes of these standards, immune effector cells are broadly defined as cells that have differentiated into a form capable of modulating or effecting a specific immune response. Donor lymphocyte infusions are considered to be part of the hematopoietic cell transplant standards because, although lymphocytes are administered for their immune effect, it is only in the context of transplantation. Immune effector cells focus on novel therapies. They include CAR T cells, T cell receptor modified T cells, antigen-specific T cells such as virus-specific T cells, NK cells, B cells, vaccines, and TILs. For purposes of accreditation, all cells administered within the program are addressed. Both research and licensed commercial products are within the scope of the FACT standards. Patient access to immune effector cell therapies, the safe administration of these therapies, and improved patient outcomes are the motivation for standards development in this field. At least 4 groups have demonstrated an interest in development of immune effector cell standards. FACT-accredited transplant programs are interested because they participate in immune effector cell trials and are using commercial immune effector cell products in their services. They have a desire to apply FACT requirements to these services for the safety and improved outcomes in their patients. Drug manufacturers are invested in a controlled and safe clinical trial and have a need for continued assurance of proper handling and the use of these products following licensure. Regulators are interested in the field's ability to handle these challenges, chain of identity and custody, toxicities, and complexities, because they are responsible for approving only safe and effective products for licensure. Payers, too, are interested in that there will be requests for reimbursement, and they have an expectation of good outcomes for the covered services. Standards for immune effector cells were developed for several reasons. Therapies were being administered on the clinical transplant unit, and experts saw the importance and need for standardization and support. These are complex therapies with many similarities to hematopoietic cell transplantation. They require an infrastructure of robust systems to ensure safety and efficacy. Importantly, cellular therapy is more than just the cellular therapy product. It includes the recipient selection, product selection, collection of the starting cellular material, preparative chemotherapy regimen, post-administration supportive care, long-term monitoring, and reporting requirements. Standards and accreditation can guide the expansion of existing quality programs to accommodate new therapy services. In addition, standards and accreditation can guide new program development when therapies are used in clinical situations outside of the transplant unit. Immune effector cell therapies are similar to hematopoietic cell transplantation, but different. Both have a need for clearly defined responsibilities among numerous participants. Immune effector cell product manufacturing is complex, unlike traditional pharmaceuticals, and is more likely to involve third-party manufacturers, clinical trial sponsors, and others. The chain of identity, chain of custody is more challenging when products are transported outside of the institution. Immune effector cell therapies also routinely involve more interdisciplinary clinical groups such as neurology, the intensive care unit, solid tumor oncologists, and treatment settings. Immune effector cell therapies may have unique and serious or fatal adverse reactions. Some of these are unpredicted and unpredictable. However, adverse events are always significant, even if rare, and when they occur, they may be a setback for clinical trials. This slide illustrates a typical clinical pathway to show the similarities and differences between hematopoietic cell transplant and immune effector cell therapy using a licensed CAR T-cell product as the example. Across the top, you see the traditional pathway for hematopoietic cell transplantation, wherein the patient on the far left is identified, a donor or stem cell collection occurs, there is treatment and infusion of a product which has been stored at a laboratory generally on site. Following transplantation, patients are followed usually by the transplant service for a defined period of time and then for a longer period of time in long-term follow-up. In contrast, along the bottom, you see the typical pathway for an immune effector cell therapy, which also begins with identification of the appropriate patient and a collection of a cellular therapy product or cell collection. This collection then is transported to a cellular therapy manufacturing site, generally off-site, although not always. The patient then undergoes additional treatment. When the cellular therapy product manufacturing has been completed, it is transported back to the site of the patient where it is infused, and follow-up of the infusion may be short-term by the infusing team, or the patient may be transferred immediately back to the referring physician. The major differences then between these two lines of therapy are variable protocols for collection of the starting material, the duration and location of manufacturing, the careful planning necessary to ensure that the patient is ready when the cells are ready, more diverse group of physicians and medical teams caring for the patient, and the location and arrangements for long-term follow-up are less well defined. So why does all of this matter? There are over 160,000 adverse patient events occurring each year in the United States because of patient or specimen identification errors. 11% of all transfusion deaths are the result of not properly identifying the patient or of mislabeling the tube of blood used to identify compatibility of the unit. The chain of identity is not a trivial issue. FACT standards require the use of ISBT 128 labeling and verification of identity of the product and the recipient by 2 qualified individuals or a validated system and 1 qualified individual. Next, I will describe a couple examples of why immune effector cell therapy standards are needed to help ensure chain of identity, chain of custody, and administration of the right product to the right patient. The label format and content varies among companies and sponsors. Companies often use study number, which is not unique and not in the patient's electronic medical record. These products are importantly different from other drugs that might be administered to the patient because we rely 100% on labeling and identifiers to ensure the right product for the right patient. There's no real-time serologic or antigen testing available to confirm the identity of the product. So in this first example, it was an autologous mononuclear cell product collected from 2 patients on the same clinical trial from the same institution. These products were labeled and sent for manufacturing. The following information represented the unique identifiers: the patient initials, the study number, the enrollment number, and the date of birth. At collection, the products were labeled as you see here with the initials, study number, enrollment number, and date of birth for the 2 patients. The CAR T-cell products returned to the institution were labeled as you see here with the initials, study number, enrollment number, and date of birth. However, the dates of birth did not match what had been sent. Despite considerable effort, neither product could be used because there could be no assurance of where the error occurred. In this example, a contract manufacturing organization required the apheresis center to label the collection with the patient name, the patient medical record number, and the patient date of birth. Without notification, the CMO changed the final product label policy and sent the product to the clinical site without these identifiers. This created an urgent situation for the clinical team as they could not use the product as it was delivered. Ultimately, the CMO was able to link the identifiers on the bag to the patient-specific identifiers from the collection bag at the manufacturing site. In this case, the product was able to be infused. So, how to connect the dots? This is what the clinical team sent to the manufacturer on the left. The identifiers shown by the green arrow are on the bottom of the label. What they received back is shown on the right with a lot number and an ID having no apparent relationship to the identifiers on the original label. The clinical team responsible for the infusion has no ability in this case to verify identity and puts its patient at risk if this chain of identity cannot be confirmed. Lessons have been learned through early experiences with immune effector cell therapies. The chain of identity and chain of custody, including labeling, are critical. Both programs and commercial manufacturers want and need greater consistency to avoid confusion and critical errors. A robust infrastructure is required to administer the right immune effector cell to the right patient at the right time with the right management and supportive care available and implemented. Clinical programs assume responsibility for quality, even though FDA approval is an important factor. Accredited programs have an opportunity to work with commercial manufacturers to effect process improvements in this area. I'll turn now to a discussion of immune effector cell standards and accreditation. FACT standards for immune effector cells are comprehensive. They include clinical activities for both IND and commercial products. They include all phases of care from recipient selection evaluation through management and follow-up, product preparation, administration, and management of adverse events, compliance with REMS as required by the manufacturer, evaluation of clinical outcomes individually and in aggregate, and reporting of outcomes to a national registry. A quality management program is essential in an accredited program. Quality management programs establish, maintain, and monitor and implement process improvements throughout the program. A quality management program includes written SOPs, personnel training and competency, documentation of record control, good documentation practices, documentation of adverse events, clinical follow-up, and long-term follow-up. A quality management program in a cell therapy setting for transplantation incorporates immune effector cell products and patients into the same system. The scope of the FACT immune effector cell standards includes also cell collection. These cell collections are hematopoietic progenitor cells or mononuclear cells obtained by apheresis and by bone marrow. The cell collection facility also requires a robust quality management program. Manufacturing, as it occurs within the accredited laboratories, is also included within the scope of immune effector cell standards. The laboratory is expected to have control of its processes, equipment, supplies, reagents, facilities, and personnel. At a minimum, the laboratory is often responsible for product receipt and preparation for administration. The focus of the standards for most laboratories is on chain of custody, chain of identity, labeling, transportation, and storage. The processes can be more comprehensively included if the facility does actually some of the manufacturing of clinical trial products. There are many clinical models wherein immune effector cells are given. The BMT unit, the non-transplant clinical site, and immunotherapy divisions or institutes are the three most common models. On the BMT unit, physician involvement is used to determine the responsibility of the BMT clinical program for immune effector cell therapies. When the BMT physicians are involved in the care and management of these patients, this is considered part of the BMT unit services, and the BMT program is responsible for immune effector cell therapy. If the physicians are not involved, but the patients are managed on the BMT unit, this is considered generally renting space and sharing resources such as nursing and pharmacy. In this case, responsibilities must be clearly defined, and it's possible this may have no BMT unit involvement in the accreditation of the immune effector cell therapies. Immune effector cells can also be administered on a non-transplant clinical site, such as a leukemia or lymphoma service, in which case a parallel system to the BMT system would be required for this independent service. The third model is that of an immunotherapy division or institute, wherein the immune effector cell therapies are administered across several different clinical sites under the directorship of the same leadership. A standalone immunotherapy program is eligible for FACT accreditation under the IEC standards. If operating outside of a transplant program, the IEC service must meet all the standards, including a robust quality management program, which is often the most difficult to achieve. This slide is a graph of the accreditation of clinical programs in cellular therapy. What you see in yellow are the currently accredited clinical programs, 184. Of these, 160 are accredited for allogeneic transplant. There are 111 accredited programs for immune effector cell therapy. Of these, only one is a standalone immune effector cell program. The other 110 are located within a cellular therapy transplant program. Earlier, I described the accreditation process as one of submission of documents, an on-site inspection, notation of citations or lack of compliance with standards, and an opportunity for programs to make correction. These are some examples of the types of citations seen in immune effector cell programs through the FACT accreditation process. They fall into two general categories. The first category is citation related to a new program, wherein the services and expectations are not fully developed, and so the citations were noted as things being not present yet in the immune effector cell program. The other category of citations are more general failures to meet standards that would be expected for any cellular therapy program. So of these examples are listed here as cited by the on-site inspector. Although the program reviewed outcome analysis and efficacy, there are no endpoints defined to assess the efficacy or safety of the immune effector cell therapy. This is undoubtedly an example of the early phase implementation of immune effector cell therapies and will result in a more mature process as the program matures. Other citations include cells not being stored at the correct temperature, being stored at the apheresis machine prior to distribution, but this process not being well defined in a standard operating procedure. In other cases, there were no specified time limits for distribution after collection, certificates of analysis examined for some reagents but not others, equipment not calibrated or maintained within the appropriate time frame, and specific parts of the apheresis equipment were not regularly cleaned. In cases where these citations are noted, they would be completely described to the program. The program would be expected to document its correction by a submission of a new SOP, a temperature log, or other form of documentation of implementation of the SOP. This response to citation is reviewed by the FACT staff coordinators and/or by the accreditation committee if necessary. When all requirements have been met, accreditation can be awarded. FACT has immune effector cell standards which address many new therapies that are under development. There are some special considerations for solid tumor cellular therapies that are not completely identified and addressed within the current edition. The next edition of immune effector cell standards is currently being drafted to address some of the gaps identified during the period of public comment in 2020-2021. Many of these perceived gaps related to tumor infiltrating lymphocytes, or TILs. Examples of things to be addressed in the next edition will be collection requirements for tumor extraction for further manufacturing, tissue handling, transport, integrity, and labeling, chain of identity, chain of custody considerations, collection at remote sites not otherwise accredited, and clinical care considerations, especially for such agents as IL-2 expertise. Other issues if the patient is not in an institution with a FACT-accredited hematopoietic cell transplant program will also be addressed in the next edition. This is expected to be open for public comment this winter. We'll move on now to quality management, REMS, and audits. All FACT standards require the establishment of a robust quality management program. Elements that must be minimally included are detailed in the standards, and these include the following. Importantly, starting with organizational structure, key positions must work together to accomplish a quality management program. Personnel, including the key positions, education and experience required, qualifications for the job, a job description, training competency, and continuing education. The third element is that of document control, which includes controlled documents, standard operating procedures, policies, worksheets, forms, and labels, all of which must be controlled in their creation, approval, implementation, revision, and archival. Written agreements with external parties that provide services impacting the cellular therapy product must be obtained and reviewed on a regular basis. All programs are expected to have a robust program for outcome analysis. Data to be included are both individual and aggregate data. Specific data points are noted in the standards for the more mature cells. All programs must meet the expected survival outcomes as measured by a national or international benchmark. When these benchmarks have not been met, a corrective action plan is required to be submitted to FACT. The quality management program must provide routine auditing schedule. The specific clinical activities to be audited are listed in standards and include such things as drug doses, data accuracy, donor screening and testing, and eligibility determination. Standards also require that there be policies and procedures for the management of cellular therapy products with positive microbial culture results. Occurrence management is an important part of the quality management program. For purposes of these standards, occurrences are defined as errors, accidents, deviations, adverse events or reactions, or complaints. All of these occurrences must have systems for detection, investigation, documentation, review, reporting, and corrective and preventive action. Chain of identity, chain of custody is part of product tracking and tracing, which is an important part of a quality management program. All programs must have a disaster plan that includes major disruptions and lesser interruptions. All critical procedures and equipment must undergo appropriate qualification, verification, and validation. The program must have a system for change control and assessment of effectiveness of the quality management program. The program is expected to obtain feedback and share the results of the effectiveness of the quality management program in order to accomplish improvements. Turning now to risk evaluation and mitigation strategies, or REMS. REMS are a portion of an overall risk management plan. They are defined by the U.S. Food and Drug Administration for drug products, including biologics, with serious safety concerns. They are intended to ensure that the benefit of the therapy outweighs the risk. All licensed drug products have required post-licensure safety measures. Labeling and packaging inserts are part of these safety measures. REMS requirements are to supplement when the product is thought to have a greater risk. The FDA determines when REMS are required. Manufacturers develop the REMS and FDA approves the proposal. The REMS system is for one product and one company, so variability among these different programs is completely expected. This slide outlines the responsibilities involved with REMS. The regulatory authority specifies when they are necessary and approves the REMS for specific products. Manufacturers develop the specific program, implement the program, and monitor compliance with the program through audits of the clinical sites. The provider enrolls patients in the therapy and becomes certified in the REMS program. The provider must comply with the REMS training, consenting, prescribing, and patient management. This includes the entire team involved in patient care, including training. This is for pharmacists, the ICU, the emergency room, and also for the collection center and laboratory as they are involved in these therapies. Patient and family have responsibilities also to understand the product risks, comply with specific requirements such as lab, registry enrollment, the location restrictions that might exist immediately following therapy. Patients are also responsible to inform other providers that they received a cellular therapy and advise on the use of other medications. Key points about REMS are listed on this slide. The FDA determines if a REMS program is needed, but does not dictate the content. The manufacturers develop the content of the REMS program, explaining why there is a considerable variability among products. The focus of the REMS is on one single product. Generally, it involves at least the following elements: designation of an authorized representative responsible for the product, training of all personnel who manage the product. Generally, the content of training is dictated to include, at a minimum, collection, shipment, administration of the one product, expected adverse events, management, appropriate drugs to treat the adverse event, short-term follow-up, reporting of immediate adverse events. And finally, communication with the patient and provision of education, often using a tool such as a wallet card for the patient to maintain the identification of the drug and appropriate warnings. Importantly, REMS are not intended to address infrastructure necessary to promote drug safety. This is an example of education requirements of the current CAR-T REMS programs. Training for a specific drug or biologic is required. Full prescribing information, medication guide, and adverse event management. Training is conducted via written materials and live training. There is a knowledge assessment, and it's required of the authorized representative and anyone involved in the prescribing, ordering, dispensing, or administration of the biologic. This includes new providers and staff, and all providers and staff if the CAR-T product has not been dispensed in a year. This is generally the retraining provision as described in the REMS programs. In contrast, education requirements of FACT for immune cell programs include the following. Experience. Physicians must have greater than a year experience managing patients with applicable therapeutic diseases. Training and competence. Physicians must be trained and competent in the program services. There's an extensive list of knowledge requirements for physicians involved in cellular therapies, and similar requirements exist for other members of the healthcare team, including physicians in training, advanced practice providers, nurses, pharmacists, consulting physicians, quality managers, data management staff, and support services. This slide summarizes accreditation versus REMS characteristics. FACT accreditation is comprehensive for cell therapies. REMS addresses a single product. Accreditation addresses continuum of care, all therapies administered to treat the patient. REMS addresses collection and infusion of a single product. FACT accreditation requirements are for broad educational experience. REMS requirements are for prescriptive management of adverse events. A minimum of 1 year follow-up is required for the accredited program, whereas short-term follow-up is required in REMS requirements. FACT accreditation addresses the choice of product most appropriate for the patient. REMS addresses one company, one product. FACT accreditation addresses the infrastructure necessary to leverage education, experience, and training to be ready for the next product. REMS requirements have no infrastructure provisions. In immune effector cell therapy, the product is only one part of the therapy. As shown here, both FACT accreditation and REMS requirements address specific risk of the product and use of the cell therapy product. However, only FACT accreditation includes treatment and management of the primary disease, specific selection of therapy, and other effects of cell therapies included in the entire continuum of patient care. In addition to requirements of REMS, most programs will be audited more than once by multiple manufacturers. Each audit is specific to a cell therapy product. Some manufacturers will conduct separate audits for different products, and some will conduct separate audits for the same product because of different research studies or different licensure requirements. The result is illustrated here. A significant amount of effort in auditing is duplicative. Each manufacturer is potentially looking at the same issues. As shown here, frequently these same issues have already been addressed by the FACT inspection and accreditation process. In addition to the specific requirements for individual products and the specific manufacturer audit points, the broader scope of FACT accreditation is apparent. Manufacturers and FACT inspectors have collaborated on inspections and made some observations of the audits and FACT accreditation inspections. FACT inspections generally cover the majority of concepts audited by manufacturers. This has been determined by a crosswalk of the FACT standards and 3 different audit checklists issued by manufacturers. Some product-specific requirements are not listed in the FACT standards and are not expected to be covered by the FACT accreditation. FACT incorporates feedback from these kinds of collaborations from industry in standards development processes to facilitate collaboration and minimize the burden on programs. There are several methods in which manufacturers are leveraging FACT accreditation in their site qualification visits and in their audits. The goal is to reduce the burden on healthcare providers, institutions, and also on the manufacturers. FACT accreditation can be leveraged whether or not the manufacturer chooses to require accreditation. The manufacturer can perform a simple desk-based audit for product-specific requirements and for evaluating performance of FACT-accredited institutions. The manufacturer can perform abbreviated audits of FACT-accredited sites. FACT provides written reports to the programs which they are able to provide to manufacturers at the manufacturer's request. At least one company is requiring the FACT accreditation report, but another is not. So the methods of collaboration and cooperation are variable. Collaboration applies to both clinical trial and commercial products. Recently, more discussion on clinical components has occurred and is greatly encouraged by both FACT and some manufacturers. Payers and manufacturers have recognized the benefits of immune effector cell accreditation. FACT is one of many stakeholders to help create the infrastructure needed to deliver safe immune effector cell therapies and cell and gene therapies. Dialogue with commercial manufacturers to facilitate productive working relationships with programs is facilitated through FACT. This provides many opportunities for greater consistency with less burden on programs and facilities. One FACT-accredited program has provided a testimonial to the importance of FACT accreditation and in its experience with immune effector cell therapies. This FACT-accredited program currently works with 8 different companies in cellular therapies. They found that accreditation preparation caused the program to put in place all the elements to best care for the patients beyond any single REMS set of requirements. Depending on the company, this program gives feedback about their processes, including chain of custody identity, labeling, controls, and even patient care. Most importantly, FACT standards are far more comprehensive than any set of REMS requirements or qualification audits. These accreditation positions the program to take on any new product, whether it's a clinical trial or licensed product, and successfully navigate the cellular therapy from collection of starting material through manufacturing, administration, patient care, management of toxicities, and long-term follow-up. This program identified accreditation elements that they have in place for adoption of immune effector cell therapies because of their accreditation. These include the following: defined leadership, broad-based training program, and regular coordination among all the services that a patient receiving immune effector cell therapy might require, such as the emergency department, intensive care unit, 24-hour phone access to attending physicians. Adverse or unexpected events must always be appropriately recognized and treated. The program is prepared to provide life-saving care even if such adverse events are rare. Additional elements identified by the program as being in place because of their accreditation include a strong quality system, including organizational structure, document control, adverse event reporting, risk management, and change management. Accreditation prepares the program to handle new products, approved and investigational products appropriately. One REMS program does not prepare the program for the next REMS, as many are quite different in completeness and in character. The program identified one of the most important elements of accreditation as the appropriate care of the patient. Disease-specific groups decide which overlapping drugs will be used in which circumstances. Standards require specific training and experience of the team members. Educated team can also provide ongoing evaluation of new products, investigational products as they become available, and they are therefore able to bring these new products to the patient appropriately and safely. Finally, event reporting and data management are simplified through involvement with the transplant program. Events from all IEC products are examined and communicated, and where needed, corrective action is taken. Events are not siloed but are shared across the program and all groups involved in the cellular therapy. FACT participates with many multi-stakeholder groups in provision of immune effector cell therapies. Many multi-stakeholder groups have formed to standardize aspects of cellular therapies as more products become licensed. Participation of manufacturers, healthcare providers, payers, accrediting organizations, data registries, professional societies, and more are strongly encouraged. These are some examples of the multi-stakeholder groups. 80/20 Task Force of the ASTCT. This task force is based on the premise that as much as 80% of the requirements for immune effector cell therapies can be standardized across products and companies. Another group is the Deloitte NextGen Industry Working Group and numerous subgroups, including site certification, long-term follow-up, labeling, chain of identity custody, and more. Finally, the standards coordinating body and numerous subgroups, including apheresis, collection, chain of identity and custody, cryopreservation, and more. This is an example of standardization. ASTCT, one of the parent organizations that founded FACT, has published a consensus on grading for cytokine release syndrome. This is important collaboration to allow good communication among providers, standardization of care, and hopefully improvement in the management of this adverse event. FACT collaborates with the CIBMTR on data management and data auditing. CIBMTR operates the Cellular Immunotherapy Data Resource, or CIDR, through a grant from NIH. This is part of the Cancer Moonshot program to accelerate cancer research under the Immuno-Oncology Translational Network. FACT requires collection and maintenance of complete and accurate data. The 8th edition of FACT standards recommends submission to CDER of all immunotherapy results. There are forms specifically designed for this. CDER gathers data on cellular therapies for all cancers, including solid tumors and non-malignant diseases as well. This is a centralization of data that allows long-term follow-up and is especially important as patients may receive more than one cellular therapy over the course of their lives. CDER Stakeholder Council includes representatives from professional societies, industries, payers, and patient advocacy groups. Some commercial manufacturers already use the CIDR database for collection and maintenance of long-term follow-up data. Hopefully, this will continue to grow and be a central resource for all involved in cellular therapy. Standardization of labeling is critical to safe use of cellular therapies. FACT requires full implementation of ISBT 128 in accredited facilities. Collection facilities are challenged with the burden of managing different types of labels and printers required by different manufacturers. A multi-stakeholder working group recently focused on the label for the collected cells, the starting material, at the end of collection. This working group included the standards coordinating body, the Deloitte Next Gen Industry Working Group, the ICC BBA, the administrator of ISBT 128 standard, FACT, Be the Match Biotherapies, and others. The group included some major sponsors of cell therapy clinical trials in the US and the EU. A draft label was published in 2020 for public review and was finalized and published in 2020. This slide shows the result of that work. The standard builds on the existing ISBT 128 standards for cellular therapy and uses compatible label dimensions. It retains the essential ISBT 128 traceability information while also accommodating sponsor manufacturer information in a standardized manner. This is an example of the clinical trial label. The design specifies both content and location for information. ISBT 128 traceability information is printed on the left side of the label. This includes the donation identification code, or the DIN, seen here on the label left side upper corner, starting with a 13-digit identifier and an associated barcode. The second element is the product code and text in the lower quadrant on the left side. It is both a barcode, a code, and eye-readable language that says MNC apheresis for further processing. On the right side of the label is the sponsor manufacturer information. It is printed also in a standardized format and location to ensure proper identification. The data matrix 2D symbol in the bottom right quadrant is ISBT 128 compound message that includes the DIN, the donation identification number, the product code, and the expiration date for the product. Use of this label should provide some flexibility and increase standardization to enhance patient safety in the field of cell therapy. There has been a lot of interest in the FACT Regenerative Medicine Interchange meeting. The second Regenerative Medicine Interchange, hosted by FACT, took place at the Facilitate Advanced Therapies Week conference in Miami, Florida, January of 2020. Participants from several organizations outlined needs to advance cell and gene therapies. They shared successful collaboration and discussed needs in standardization, workforce development, and advocacy. The meeting included numerous participation from professional societies, accrediting organizations, investigators, healthcare providers, industry, data registries, and more. A summary of this meeting has been published in Cytotherapy in Advance online and soon to be in print. Finally, I will describe the FACT Consulting Services. FACT Consulting is a subsidiary of FACT and offers consulting services provided by experts in the field of cellular therapy to assist the client organizations to achieve quality improvement, comply with accreditation standards, and achieve readiness for FACT inspection. FACT Consulting Services are completely independent of the FACT accreditation services. This is a fee-for-service resource that can be tailored for specific needs. Consultants are excellent practitioners, familiar with accreditation standards and accreditation, and have averaged more than 10 years of experience working with FACT. Services provided include the following: compliance assessment, review of policies, development of policies, assistance with assembling application for accreditation, mock desktop review of application materials, a mock on-site inspection, assessment of continuous FACT readiness through gap analysis, quality management assistance, training workshops, and any customized services as defined by the applicant. Additional information about FACT consulting services can be found on the FACT website. Thank you for your attention.